Imagine a world where shedding pounds doesn't require grueling diets or invasive surgeries. Sounds too good to be true? Well, scientists at the Salk Institute have just uncovered a tiny molecule that could revolutionize how we approach weight loss. But here's where it gets controversial. This breakthrough isn't just about slimming down, it's about tackling obesity, a global crisis that's doubled in the last 30 years, affecting over 1 billion people and fueling metabolic disorders like diabetes, heart disease, and cancer. Current treatments, from lifestyle changes to GLP-1 drugs like Ozempic, often fall short, leaving patients struggling to maintain results or access care. So, what's the game changer? Microproteins, a largely overlooked class of molecules hiding in plain sight within our bodies. And this is the part most people miss. These microscopic powerhouses could hold the key to treating obesity and its deadly companions. In a groundbreaking study published in Proceedings of the National Academy of Sciences on August 7, 2025, Salk researchers used CRISPR, gene editing, to screen thousands of fat cell genes, uncovering dozens that likely code for microproteins regulating fat cell growth or lipid storage. One of these, confirmed by the team, could become a drug target for obesity and metabolic disorders. This isn't just a scientific win, it's a testament to CRISPR's potential to reveal hidden biological regulators. As senior author Alan Sagatelian puts it, the more we screen, the more disease-associated microproteins we find, and the more potential targets we have for future drugs. But here's the kicker. While microproteins like GLP-1 have shown promise, they're not without flaws. Side effects like muscle loss and nausea have left patients and doctors searching for better options. Could the newly discovered microproteins be the answer? Sagatelian's team thinks so, and they're using cutting-edge CRISPR tools to explore the dark corners of the genome. Regions once dismissed as junk DNA, these areas, now known to harbor a hidden world of microproteins, are expanding our understanding of protein libraries by up to 30%. CRISPR screening works by cutting out specific genes and cells and observing their impact. In this case, the Salk team focused on genes tied to fat cell development and lipid accumulation. We wanted to uncover what we've been missing in metabolic research, explains Victor Pai, the study's first author. CRISPR lets us pinpoint genes that directly influence fat storage and cell growth. Building on previous work, the team expanded their microprotein database using a pre-fat cell model that captures the entire differentiation process. They then screened these cells to identify microproteins involved in fat cell biology, narrowing the list to 38 candidates linked to lipid droplet formation.
Here's where it gets even more intriguing.
One of these microproteins, adipocyte SMORF1183, has been verified as a key player in lipid accumulation. This discovery not only validates CRISPR's effectiveness, but also opens the door to new obesity treatments. But don't think this is the end. The team plans to repeat the study with human fat cells and hopes their work inspires others to explore the dark genome. After all, as Sagatelian notes, research is a constant process of improvement. So, what do you think? Could microproteins be the future of obesity treatment? Or are we overlooking potential risks? Will CRISPR continue to unlock biological secrets? Or are we biting off more than we can chew? Let's spark a conversation. Share your thoughts below.